Okay, good morning everyone and welcome to the 2013 Royal Tyrrell Museum Speaker Series. Today the Royal Tyrrell Museum and its cooperating society are proud to present Mr. Benjamin Borkovic. Ben is a master's student in the Department of Biological Sciences at the University of Calgary. Growing up in the Northwest Territories, Ben developed a passion for the natural world and went on to study geology and biology at Queen's University in Kingston, Ontario. Upon completing his bachelor's degree, he returned to Yellowknife to work for 2 years before deciding to pursue his lifelong interest in paleontology at the U of C. For his master's degree, Ben studied ceratopsians and mammals in order to determine if horned dinosaurs showed evidence of a difference in horn size and shape between the sexes. Ben successfully defended his thesis before Christmas and is now working on publishing the results of his research. Today, Ben will present an overview of his research in a talk entitled Searching for the Sexes in Dinosaur Horns. So without further delay, I present you Mr. Benjamin Borkovic.
Thank you, Francois. And thank you very much, everybody, for coming. I hope you all can hear me. So as Francois mentioned, I just finished up my master's at the U of C, where I worked with Dr. Tony Russell. And I'm really happy to present what I was working on for you guys today. So I was looking at, as Francois mentioned, sexual dimorphism in dinosaur horns, so in the horns of ceratopsid dinosaurs. And to start out, the thing that got me interested in this topic sort of came from a few observations I made in the fossil record. The first is that, sorry, I got all these tools here. If you look at the skulls of ceratopsid dinosaurs, they ended up evolving the largest skulls of any land animal of all time. And that's a really interesting feat. And both in absolute size, so overall, just the length of the skull, and in proportional to their body size. And so this is sort of indicates that they, well, I'll come back to that, but yeah, they had very, very large skulls and the largest skulls of any land animal ever. Second to that, if you look at the ceratopsian phylogeny overall, as you move up through time and through size, there's a steady enlargement of the skull relative to body size. And so combined, both of these signals sort of suggest that there was a continuous selection on the skulls of these animals to increase. And that's sort of an interesting signal. In addition to that, if you look at the diversity we see in ceratopsid skulls, there's a great deal of variation in the frills and in the spikes and the horns that adorn their skulls, but relatively little morphological variation in the actual skulls themselves. So collectively, this sort of suggests that there was strong selection going on for enlargement of the skulls and for what the skulls displayed. And so a lot of people have talked about whether or not this might be as a result of sexual selection or if it's something more like species recognition. And along with this, a lot of paleontologists have suggested that there's probable sexual dimorphism that goes in these dinosaurs, and that would just sort of follow suit with the theory of sexual selection. But there's actually very little evidence to support that. And a few people have looked at it, but nothing's really been concluded thoroughly. So I thought maybe it was time to have another look. So what does it matter really if the horns of these animals are or are not dimorphic, if they're larger in males or larger in females, or if there's a difference in shape? Well, it has a lot to do with how they lived. And the morphological variation that we find in a species, whether it be in their skull or in their body or in some other feature, can actually tell us a lot about the selection pressure that led to that sort of end form and how they got there. And in the case of ceratopsids, we actually know relatively little about how these animals how their horns were used sort of in day-to-day life and under what kind of context they evolved. If you just Google Triceratops, the very first thing you'll find is a picture of a Triceratops fighting a T-Rex. And this has sort of been portrayed in 101 different ways. But we have very little evidence that this sort of classic showdown ever really took place. Surely Tyrannosaurs ate Triceratops, but with a lot of discussion about whether or not they just scavenged. It could be that they only ate them once they were dead. It's probable that they fought, but there's actually little sort of bone-on-bone evidence for this kind of behavior. The other scenario that's often portrayed is sort of, you'll see two bull Triceratopses fighting over dominance of the herd or in the watching eyes of some female. And again, there's little evidence for that. People have started to look for it. There's been some interesting studies trying to look at bone lesions and their placement, but relatively little evidence to suggest that this actually happened. And if we were able to nail down definitively that there is or is not a dimorphic pattern in these horns or in the frills of these animals, that actually might tell us a little bit about whether or not they're used in these sort of contexts. Before I go any further, I'll just explain what I mean by sexual dimorphism. And by that, I'm just referring to morphological differences between males and females that are of the same ontogenetic stage, or basically the same age. And because we're looking at morphological differences, I would argue that those are things that need to be sort of being demonstrated statistically if it's a quantitative trait, or quantitative trait, or if it was qualitative trait, something you can notice directly. And I'll use the term ontogeny a lot, and basically that just refers to the development of the organism, and if you think of it as growth, then that'll be more than enough for now. So there's actually a lot of evidence in the fossil record for sexual dimorphism, and this is a topic that's been discussed lately, and I thought I'd just highlight some of the key or most interesting cases that I came across in my research. And there's a great deal of fossils that came out of the Bear Gulch limestone in Montana, and they were all sort of studied by Lund through the 80s and 90s. This is a really good example of Falcatus falcatus, where you see the male has this dorsal spine that projects over the skull, and two specimens were actually found in this pose with the spine from the male in the mouth of the female. And so it's fairly good evidence that this is a dimorphic trait, and it's sexually dimorphic, so that this would differ between males and females, and that it was probably involved in copulation. How, I don't know, but there's some sort of grasping involved. Similarly, in a holocephalian species from the Bear Gulch again, this is called Harpago feutator. And as you can see here, the female specimen looks reasonably normal as far as fish go. And the male has these huge paired clasper things that project out of off the ethmoid plate on the skull. And they're segmented, they're heavily calcified, and they're quite large all over the skull, and I have no idea how these were used. But it's a really, really interesting case of sexual dimorphism. And something that I'll talk about in a moment is that, this could just be two wonky species, or two species that look similar, and one has this sort of elaborate cranial feature and the other one doesn't. But the real advantage of working with chondrichthyans, and why there's been so many interesting examples, is that in the modern day, if we look at sharks, they have these pelvic claspers in males, and these develop over the course of the male's life and become fully formed at sexual maturity, and as the males age, they calcify. So they're actually the sort of thing that will stick around in the fossil record. And luckily enough, chondrichthyans have had these since at least the Devonian. So basically, the majority of the chondrichthyan fossil record, you have kind of a dead ringer that gets preserved with most of the skeleton that'll tell you whether or not it's a male or a female. And so from there, even if we didn't have some sort of crazy feature like this, you can start to look at the differences between males and females right away. And it makes it a lot easier to sort of work out the statistical differences there. There's a lot of other neat cases as well, many, many instances of sexual dimorphism in mammals. This is a early rhinoceros from the Miocene, and there had been a huge number of these specimens found in Nebraska, and people were fairly certain that they'd nailed down male and female differences. But the real sort of clincher was when they found inside of the pelvic bones of what they thought was already a female, they found the fetal remains of a embryo, I guess, or a fetus in the pelvis of the female, and that kind of made it definitive that that is indeed a female species, and the dimorphism they were noting was indeed sexual. Just a couple of years ago, this species of Darwinopterus was found, or this specimen of Darwinopterus. And again, this is a case where there was an expectation of dimorphism. People had already looked at a few specimens, and there seemed to be a few differences between males and females relative to related to the presence of a crest on the skull and the size of the pelvis canal, or the pelvic sort of egg canal. And then this specimen was found. And just right here, directly outside of the where the egg would be laid, they found an egg, basically. And the thinking is that this specimen died, fell into the water, and as the body kind of began to decay, the egg slipped from the birth canal and ended up being preserved directly in association with it. And it would be really, really unlikely that an egg rolled from somewhere else and happened to have stopped right there. So this is fairly good evidence that this was a female specimen. And then again, this sort of confirmed what their previous expectations were of the female morph. One of probably the best one, just came out this summer, actually. And these are turtle specimens of, I believe, Aleocheles from Messel. And there's 9 copulating pairs have been found. And all of them share a similar size difference and pattern. So people are very, very confident to say that this is the male specimen, and these are females. And females are about at least 20% larger in carapace size. They've got a shorter tail than the males do, and there's a difference also in some of the hinging in the plastral plates. So a really neat case, and this is, I believe, the first record in the fossil record of animals that have been preserved sort of in the act, so to speak. Probably the best case, though, in terms of illustrating examples of sexual dimorphism comes out of some of the pachypleurosaurs from the Triassic of Europe. And people had noted an interesting difference in the humeral size and shape since about the 1920s, but it was finally really sort of studied and published on in the 80s. And so these marine reptiles, which would be secondarily marine, people were noticing a difference in the size and the shape of the humerus, and there was 2 consistent morphs. Furthermore, when you actually start to take measurements of them and compare them to things like the femur or other trunk size, there's this bimodal pattern. And this is something that you would look for if you're looking for dimorphism in a species, or just in the fossil record in general. So people looked at this and said, okay, cool, there's 2 morphs, there's consistent difference in body size and in humeral shape and robusticity, and the larger specimens have a stronger, more robust humerus. And so the theory kind of was suggested that maybe this was similar to leatherback turtles, and the females were the specimens that had the larger body size, and this would be based on their ability to give birth to a larger clutch of eggs, and the robusticity of the humerus would be related to their ability to pull themselves up onto the shore to lay those eggs. Of course, that got thrown on its head when people started finding neonates in the water, and furthermore, they found some inside of the body cavity of one of these specimens and realized that they don't lay eggs at all, they give live birth. And furthermore, that the specimens that they thought were male, the smaller ones with the weaker humerus, were the ones that actually had the neonates within the side body cavity. And so that was, again, fairly definitive evidence that those morphs are sexual and that the smaller one there is female. Although those were neat to study and sort of very informative examples of dimorphism, I was more interested in looking at studies that had good methods and thorough methods that I could sort of draw from when I was trying to build my project. One good example was Chris Bennett's study of Pteranodon, and he looked at several hundred specimens from the, I think, Smoky Chalk Formation, and he ended up recovering these very, very clear bimodal patterns. And in association with that, he noticed that there was differences, a consistent difference in presence or absence of crests on the skulls and in the relative size of the pelvic canal. And furthermore, he found that the presence and absence of the crests and the differences in the pelvis were associated with these size clusters. And so the smaller bodies, the more numerous, smaller size class also tended to lack the cranial crests and had a larger, broader pelvis proportional to their body size. And so that it's strongly suggested that these are female and that the larger morph are male. And you can see the reconstruction here of a full-sized female and male specimen. Another good example, as I mentioned, there's many, many, many examples in mammals, but this is, again, a Miocene rhinoceros, I think, Menoceros, I'm not sure, I can't remember. But when measuring various properties of the skull, you can see that this recovered, for nasal width against nasal thickness, a very, very clear bimodal pattern in the size of that bone. And that would be related to the presence of a horn on the snout. Just last year, Jordan Mallon gave a talk here about the work he had done looking at Anchiceratops, and that was another sort of good example of a study of dimorphism. And he took a sort of multivariate suite of measurements on the skulls of several specimens of Anchiceratops and looked at them. One thing he did was conducted principal components analysis and just look at how sort of all of the specimens relate to each other when you consider all of the variables he looked at. And he was looking for a pattern of clustering here in the over the principal components axes. And then he also looked at the examples with the cluster analysis to see if he could recover sort of a deep bifurcation in the group, which might suggest that there was 2 consistent morph patterns. And Jordan didn't find evidence to support dimorphism, but his methods were good and something that I wanted to emulate. So from my sort of initial review of the literature, it became really clear that sexual dimorphism is something that's actually quite abundant in the fossil record. It's just that it's really hard to demonstrate. And you either sometimes you end up with these really exceptional fossils and make it really easy, but otherwise it takes a lot of legwork and a lot of samples. And the 3 biggest things that will confound this work is, I guess, the first thing you have to do is sort of look for a pattern of dimorphism, look for sort of a pattern of bimodality or something that would suggest there's 2 groupings, and then you need to rule out whether or not you're actually looking at 2 different species or if it's a single species. You need to look at whether or not they're from the same age range or if you're just comparing an adult specimen to a juvenile specimen. And then you also need to make sure that they're from the same stratigraphic interval and you aren't comparing something that's from 10 million years later. And that's happened a lot, particularly in dinosaur research. And one of the examples was in some of the hadrosaur dinosaurs here in Dinosaur Provincial Park. Looked like a good case of male and female differences, and then people realized that, well, they actually occurred about 700 or a million years apart in the fossil record, so it would make getting together and reproducing pretty difficult. Furthermore, from my own work, I realized that maybe a variety of methods would be the best approach to tackling this, so I pulled several from the literature, and that an ability to compare things with an extant animal, similar to what I was talking about in chondrichthyans, would be really informative. So these are just the methods that I wanted to use for my research, and for our purposes here, basically wanted just to look at the nature of the distribution in the data to see if there's evidence of possible bimodality or multimodality. I also wanted to just look at plots of the data so I can inspect it directly, frequency histograms like in Pteranodon or in bivariate plots like in the rhinoceros example. And I also wanted to use principal components analysis and cluster analysis like Jordan did. And then one other thing I wanted to add in was to look at eigenshape, or basically just look at shape variation. That's something I wanted to do particularly in Triceratops. So I'll talk a little bit more about that later on. So from there, I wanted to look at the horns specifically in ceratopsians because of the variation that we see in there, and because there's sort of these elaborate and very large accessory features on the skulls of these animals. And also because they're a little bit conservative in terms of variation, so it's sort of easier to compare them between specimens. And the main things I wanted to look at was Centrosaurus apertus and Triceratops, and particularly Triceratops horridus. But the first question I needed to tackle then was, what was I going to use as an extant comparison? Because I needed to have something I could compare against and something on which I could sort of base my study from the modern day. So a really immediate example, or the first thing I think of really would be rhinoceros. They have a nasal horn. They look a lot sort of like Centrosaurus. They're large, graviportal herbivores. They seem to sort of be a really good example. Unfortunately, the horns in rhinos are made of keratin. It's effectively sort of the same material that your fingernails are made of. And so they're not anatomically similar at all to the horns of dinosaurs. And furthermore, for some reason, it's apparently really hard to get your hands on rhino horns. So it sort of became a huge impediment to being able to actually get a sample that'd be worth looking at. So they weren't a very good comparator. Another thing would be maybe be some chameleon species, like these Jackson's chameleons here, which frankly kind of look like little triceratops that live in trees. Unfortunately, they too are also hard to come by, and the size difference, I mean, we're talking about orders of magnitude now. And I believe there's a jar of them up at the collection in the U of A, but I'm not sure if there's anything, any more available than that. So again, it wasn't really effective for my purposes. So I needed something that I could make sort of a proof of principle study out of, and I needed to look at horns. I wanted to find something that was reasonably anatomically similar, and that would allow me to build a data set similar to what I was expecting to get in horned dinosaurs. And basically I ended up selecting bovid mammals for anatomical reasons. The way they grow horns with a bony core and a keratin sheath is very similar to how people think the horns in horned dinosaurs grew. The way they use their horns has been really, really intensively studied, and that was a huge part of the literature I'd been reading about, was about sort of the selection pressures that have led to the presence or absence of horns in various species of mammal, and also how they use their horns and how that has sort of influenced the shape or the form. And so this would be a very sort of important literature to draw from. And also for methodological reasons. Basically, they're readily available. They're fairly sturdy, and they're accessible in collections here in North America, which made life a lot easier. So I ended up selecting mountain goats. And one of the most important things about mountain goats was that males down here and females have not just slight dimorphism in size, but they also have dimorphism in their horn shape. And you can see that the horn of the male curves more steadily along the entire arc. It's a more continuous curve, whereas the females have sort of a straight base and a distal crook. And this is a good example of a female horn here. And so that would provide a mechanism by which I could test out those shape difference methods. I also selected bison because they're large and readily abundant, and they too have dimorphic horns, with males having stouter and larger horns than females. And I also picked pronghorn antelope, which are not bovids, but I added them to the study because they have very dimorphic horns, although there's almost no difference in body size between males and females. And they're very interesting, example of horn use and evolution, and they're also readily abundant here in museums in Alberta. So I ended up picking basically 3 ruminant artiodactyl species. Between the 3 of them, there's a variety of sizes, or a variety of sexual size dimorphism, as well as horn dimorphism, which would be important. And they also have differences in how they use their horns. And finally, mountain goats had differences in shape, and that was important as well. So I won't bore you here, but basically I roamed about the countryside visiting museums and measuring all of the skulls I could get my hands on for all three of these species. And I measured them in ways to try to capture as much of the major variation in size and shape that I could in their horns. So for instance, mountain goats, I measured the length of the sheath along the outside, the anterior surface, and the posterior surface, as well as the cord. And then I measured the length on both sides of the horn core as well. And then I also took measurements around the base of both the sheath and the core. And in each case, I took some measurements as well to use sort of as a body size correlate. In bison, I took similar measurements of the horn and horn sheath and of the skull. And in pronghorn, I measured both the core and the sheath as well, and tried to capture as much of the variation I could between males and females. After measuring all of those, the next thing I needed to do was go through and sort of age the sample, because I needed to make sure, like I said, that I wasn't comparing juveniles with adults, and sort of needed to come up with a way to do this for each different species. Mountain goats, it's really easy. It's basically after the first year of growth, in their second winter, horn growth ceases. So if you have the sheath in place, you can just count the annuli that they form, these rings, and then add one, and that'll actually just give you the age of the animal. It's really quite simple. If that wasn't there, I was able to look at the molar eruption patterns, and I could tell at least the cutoff between 3-year-olds and younger or older, and that was all I needed really, because anything above 3 years old is considered to be a sexually reproducing adult in the mammal literature, and so that was good enough for my purposes. For bison, I looked at the pattern of sutural fusion and the development of the rugosity in the horn and in the orbit, and that allowed me to form sort of a mature and an immature bracket and include all of the sort of adult species in there, or specimens in there. And in pronghorn, again, they have a bit of an interesting pattern of tooth eruption, but you can track it down to distinguish, again, between 3-year-olds and younger. And as long as they have a full adult dentition in place, you know that they're at least 3 years of age and therefore are actively participating in the rut. And that was the criteria I needed to get. So I won't, I'll show you some information slides now, or some data slides, and I won't worry about explaining all of it to you, but basically the first thing I needed to do with all this data was to just do sort of statistical tests to try and figure out where differences actually existed between males and females. So at this point, I separated out the males and the females that I knew, as long as they were adult, and I compared between them and just used t-tests to figure out what actually had a significant difference, and whether horns were significantly larger in males or in females, or vice versa. And so this sort of flagged for me all of the traits that, or the variables of the skull and of the horns that were different between the sexes. For instance, the basal length of the left horn, the basal width, and the basal circumference of the left horn sheath. So I was able to did this for each of the 3 species, and that gave me kind of my background against which now I could test out methods to see if I could recover those same differences. So basically, I just sort of looked at the variables, and I went through kind of all the methods I wanted to do and just see what kind of results I got. For instance, here from looking at the basal length of the horn core, you can see I recovered a bimodal pattern where I was expecting to find one. That was good, and this just gave me a chance to sort of see that these methods actually work. Whereas instead, if I was looking at the posterior length of the horn core, you're not getting that sort of a bimodal distribution, and there's no evidence in this case of dimorphism. Or again, in a bivariate plot, you can see that we've separated out a reasonably good clustering of females in the lower and males up here. And that's the kind of pattern I was hoping to see and wanted to recover. I also looked at the principal components analysis, tested out the cluster analysis, and this showed again, I was hoping to recover sort of a male-dominated cluster and a female-dominated cluster. And then there's this one little group off here, but generally it's showing that, you know, if there are patterns in the principal components analysis, you know, this grouping, and this grouping are what are popping up here. So show that these methods work and they can recover bimodal patterns or patterns of dimorphism in the fossil record or from skeletal material. The next thing I did also was to look at shape in mountain goat horns. So I traced the anterior surface of the horns in about 30 of these specimens, and then you put it into a program and it basically recovers, again, sort of a principal component scatter, but in this case, what it's separated them by is variation in the shape of that curve. And you can see that it worked out reasonably well, and I had sort of a female-dominated cluster over here along the positive aspect of the first axis, and males over here in the negative. And the neat thing was that if you move from this side to this side, what you actually get, because it'll give you an output of shape, is a change from this sort of upright with a distal crook shape that's dominated by the females into sort of a steadily curving male shape, which is what I was hoping to recover. And this showed me that this method actually does work, at least teasing out shape variation if it's present in horns. So I was able to confirm the dimorphism I was looking for in each of these mammal species. This told me that those methods I was using were working and were appropriate for this sort of problem. I was then, basically from that allowed me to sort of turn my attention then to dinosaurs and to do what I could with dinosaur horns and use these same methods. So as I mentioned, the first species I looked at was Centrosaurus II. And so these, I'm sure everyone from here is quite familiar with Centrosaurus, but basically they date to about 75 million years ago. They're found in the lower 28 meters of the Dinosaur Park Formation, which is extremely useful because that provides a restricted stratigraphic range from which these samples came and gives a little bit of a incentive to sort of refine the taxonomy there. They have a single large nasal horn in the middle of the skull, and that's what I was measuring. And they're also found in large bone beds. They have the convenient habit of dying en masse, which suggests or provides a little bit larger sample size than you might normally get from dinosaurs. And so that was part of the reason why I was so happy to work with Centrosaurus. And again, from Jordan's work, you can see here, this is the distribution of Centrosaurus apertus in the Dinosaur Park Formation. And this restricted stratigraphic distribution helped me to sort of nail down some specimens and to be sure that I was working with Centrosaurus. I also needed to look at relative aging in these groups. So luckily, just before me in Dr. Russell's lab, Caleb Brown did some work looking at, and Michael Ryan also did some work looking at patterns of bone texture transformation. And similar work has been done as well by some researchers in the States, including Alison Tumark and Duratsian and some others. But basically what they're showing is a progressive and consistent transformation in the bone texture where it changes from this sort of long striated texture into a modeled adult form. And although the actual mechanisms and reasons may not be super well understood, this provided sort of a relative aging system, and so I could look at skulls at least. And basically Caleb had gone before me and determined which were not considered to be adult. And this was useful because I needed to work only with adult specimens. As well, Centrosaurus has a rather unique pattern of horn core formation where that single nasal horn actually forms from two separate deminasals, and they fuse along the midline surface. And it's thought that this process of fusion begins as they begin to attain sexual maturity and is fully fused as they become adult. So you can see here that these two horns, this is actually the same horn just split down the middle naturally, and so it'll fuse along this face as the animal grows. And by the time it reaches adult, any evidence that suture is completely obliterated and remains only on the ventral surface of the nasal. And so as long as the nasal horn core is fully fused, this suggests it's an adult, at least as far as we can tell. And this meant that I was able to use and measure some of the disarticulated nasal horn cores that are here in the collections, that otherwise you might not know if they were adult or not. So that was a really, really beneficial component. So basically I took a series of measurements of the horn, the anterior and posterior heights, as well as the chord, and dimensions of the base, the circumference, the width, and the length. I also measured the basal skull length and the length of the maxillary tooth row, as well as the occipital condyle, which is the sort of softball-like ball at the back of the skull that keeps the head on the vertebrae, or on the spinal column, basically. And so this has been used in Centrosaurus, or in Ceratopsians before as sort of an indicator of body size. So I measured that on these as well. And I was able to look at about 10 skulls and 20 isolated horn cores to build up my data set for this. The second group I looked at was Triceratops. And I was originally going to look specifically at Triceratops horridus, but came across several specimens of Triceratops prorsus, and I measured them as well. Because, well, first of all, I'm not going to turn down a good Triceratops skull. But also the two species model is actually fairly recent, so I wanted to look at them to see if there are indeed differences between these species, as has been suggested. And I also thought that maybe if there was a sexually dimorphic signal in one species, they're so close in age, so close in time, and I mean they're so close morphologically that the difference between them has only really sort of worked out in the last 10 years, that that sort of signal may be duplicated between both of them, in which case including both species may not affect my data. They're from the latest Cretaceous, living in the last sort of 2 million years of the age of the dinosaurs, and are very abundant. However, they're recovered in isolated specimens, not coming out of bone beds. And this is a specimen, the prorsus specimen at Yale, which is a very nice skull. So I traveled around and got my hands on as many of those as I could. And one of the advantages of working with Triceratops is that there's a reasonably well ironed out growth series as they progress from sort of very juvenile stages up into the adult stage. And you can see that there's sort of a series of progressive changes relating to enlargement of the frill, the fusion of these epoccipitals along the posterior margin of the frill, and the growth and deflection of the horn cores from sort of an upright or recurved state to a procurved and anteriorly directed state. As well, the sutures all along the face, the facial region, and the frill fuse up. For anyone who's been keeping up on the Triceratops-Torosaurus debate, where Torosaurus now becomes sort of the largest super mature form, I think that's still sort of being slugged out in the literature, and so I didn't include Torosaurus as Triceratops. I was content to work with these stages, and I didn't include Torosaurus in my data. This is an example of Triceratops at the Royal Ontario, at the Royal Ontario Museum, and you can see that these open facial sutures, the reasonably short horn cores, and sort of all of these unfused epoccipitals make this into a very, very nice but subadult specimen. So again, I measured several aspects of the horn, ventral curvature, the dimensions at the base of the horn, the length of the skull, length of the tooth row, and the occipital condyle. Unfortunately for Triceratops, I had initially started out thinking I could measure the dorsal length of the horn as well. But as fusion progresses, you lose the ability to distinguish a consistent sort of stopping point for that, so I wasn't able to use that. And I looked at 12 skulls and 9 isolated horn cores, most of which were at the Smithsonian and were kind of a nice little find. When I was looking at isolated horn cores, the first challenge, of course, was to figure out which way was up and which way was left or right. But basically, if there was a remnant of the orbit, then you could actually do this, and you could tell that this was, for example, a right horn core. And as long as that junction between the horn and the orbit was present, I could measure the ventral length and be confident about where I was working from. So that was, I was happy to find those. So to get down to it, basically the first thing is that when I looked at Centrosaurus, there was elevated variation coming out of several of the horn dimensions, which I was happy to see. Although when I looked at the actual sort of distributions of the data, I wasn't finding sort of a clear bimodal pattern. Instead, I was just noticing reasonably normal distributions or skewed distributions in some places where smaller or larger individuals were more favorably preserved. But it was nothing particularly dramatic. So I moved on to looking at the sort of bivariate plots and distributions therein. And the first one, so sort of note, was if I looked at the occipital condyle height against the width, you saw there's general steady progression with size, so that was good. But one of the interesting things that I noticed was sort of 5 of the 6 largest specimens were all specimens that had a procurved horn, or a horn that angled forward. So the nasal horn in Centrosaurus is often found to basically grows up and curves backwards, which is recurved. It'll sort of grow and just stay straight, or it'll angle forward, and sometimes quite dramatically. And those are procurved specimens. And I made note of the differences, but I wasn't really expecting to find any sort of morphological differences associated with that. But that was one of the things that I found. And here, if I looked at the average height of the horn against the basal width, there was this sort of really obvious segregation of specimens with procurved horns, which are here in purple, against those that had recurved horns here in blue. And so that really caught my eye, and it's not something I was expecting to find. And when I looked at it more directly, this is basal width now over length at the base of the horn. I looked at this and did a quick statistical test, a t-test, to see if procurved horns or recurved horns were any different from straight horns, and neither of them were. But it did turn out that there was a significant difference between specimens that had recurved horns and those that had procurved horns. And Centrosaurus that had horns that angled forward actually had a significantly larger basal width than those that had their horns that angled backwards. And this really was surprising and quite interesting. On that same plot, if you get rid of those convex hulls, you can see that there's also a gap here. And this too caught my eye. And what it looked like was sort of a much more common smaller cluster and a slightly more rare larger group. And this is exactly kind of the pattern I was hoping to see. It's quite loose, but it's far from definitive. And I wouldn't say that this is really a clear evidence of dimorphism, but it is the type of evidence I was hoping to find. And this is really sort of tantalizing, this gap. But whether it's just a sort of a gap in the data, I don't know. But it's at least what what I was hoping to look for. And so this I found quite intriguing, and it'd be nice to see if, as more and more specimens can be found, if there is sort of a pattern for a larger size group. And of note, it had specimens that had straight, recurved, and procurved horns, suggesting that they're just larger overall. And if you put the labels on here and looked at a cluster analysis, there is some support, a little bit of support for that in this clustering dendrogram. And you can see that this sort of deep branch here forms this cluster group, which is the main group up here. The bulk of the rest of this grouping is all of this here, and then there's this one guy way off in the corner. But at least suggested that I wasn't just seeing things. If you stay up really late working on your thesis and stare at these diagrams long enough, you see whatever you want. So I was happy to see that there was, a little bit of maybe support for this sort of a clustering pattern. When I looked at the principal components analysis of Centrosaurus, basically there was sort of a size-based distribution again. The first one I used only skulls, but there was relatively few of them, so it was hard to read out of. But in the second one, I reduced the number of variables I was looking at and increased the number of specimens because I used only horn cores. And this here, you can see again that there's sort of a separation between, not perfect, but general separation between specimens in purple, which are procurved, or specimens with procurved horns, and those in blue, which had recurved horns. So again, this was supporting the idea that they had, and what this is really showing here, this separation over the second axis is related to an increasing stoutness of the horn, so an increase in basal dimensions with a shortening of the length. And that just showed that those specimens with procurved horns do indeed have sort of a larger basal dimension. And that was neat to see it sort of corroborated. So what I was seeing in Centrosaurus then was possible bimodal clustering in some of the basal horn dimensions. It was what I was hoping to find and tantalizing, although I wouldn't say that it's anything definitive at this point. And I also noticed that a lot of the variations seem to be associated with curvature, and this is something I was not expecting to find, so it was quite interesting. Basically, that procurved horns had a significantly greater basal width than those that had recurved horns, and also that they curve more strongly, although that's quite obvious. As I mentioned, 5 of the 6 largest specimens I looked at had procurved horns. And so this sort of was really interesting and raised some questions that I had about size and maturity. And many people before me have also talked about the procurved horns in Centrosaurus. Some people have suggested that they are more mature, that in the end, sort of as an individual grows, if it's allowed to grow long enough, its horn will sort of deflect forward and attain that procurved state. And unfortunately, with the way I looked at it, I couldn't answer the question of maturity, but I could talk about body size. And I couldn't talk about maturity because all of my specimens were considered to be mature, and that was something beyond my control. But I was looking at body size as well and size in general, so something I could at least consider. And either procurved horns are or are not associated with body size, and the evidence that I found there suggests that they may be, in which case the next sort of question is either they are more mature or they're not, which is really hard to decide, and I can't really answer that. But the other question is whether there is one growth trajectory or two. And by that I mean sort of one growth trajectory where an individual grows from those sort of 2 unfused nasals, they fuse, they grow straight up, and then they become straight or sort of recurved in the adult state, and then as it becomes very more mature, the horn eventually angles forward. Or the other option is that basically from that juvenile state, horns either grow into a recurved adult state or they grow into a procurved adult state, and that sort of depends on the individual. And I'm tempted to think that that's a little bit more parsimonious, that it's probably more likely to see just 2 sort of differing growth trajectories or a variety of differing growth patterns than to see sort of one that involves reorientation of the horn through growth. Although I don't have evidence to talk about that at this point, I find that really interesting and think that as sort of better understanding is gained about what really is going on in sort of the adult state of Centrosaurus and what it means to be actually mature, and whether we can get into this with histology of the horn cores or not, I don't know. But that's sort of an interesting avenue, and I think once that's nailed down, it'd be interesting to revisit this. And until that is more thoroughly understood, I don't know if I can say anything more about it. Basically, the one major thing, though, is that there's sort of not enough evidence to really reject monomorphism, although there's some tantalizing hints that there's something going on here that we don't fully understand. In Triceratops, again, there was elevated variation in the horns of these dinosaurs, as I was hoping to find, and also in the occipital condyles, which is a little bit weird. Most of the histograms I looked at or plots didn't reveal any sort of dramatic patterns of bimodality. but this one did, and this was the occipital condyle circumference. And this really surprised me because it's been looked at before, and typically Triceratops has a quite nice bell curve, and there was not evidence of this. So when I looked more closely, I noticed that there's sort of a separation here on the graphs, and because I included 2 taxa and also subadult and adult Triceratops horridus specimens, that I think is where this is coming from. And it seemed to be that we had sort of adult prorsus specimens clustering down here with a few of the subadult horridus specimens, and then the upper range is being dominated by adult Triceratops horridus. And so the way these are coded here are that blue squares are Triceratops horridus, red crosses are Triceratops prorsus, black specimens or black dots are ones that were not necessarily identifiable to either, but are just sort of Triceratops SP. And then I've also put S's on the ends and A's on the ends of subadult and adult Triceratops horridus or horridus specimens when I was certain of one or the other. So the bivariate plots when I looked at turned out to reveal just sort of typical growth patterns or interesting growth patterns, but there was some neat information here that I was surprised to see. The first was that just generally as occipital condyle size increases, i.e. as body size increases, we see a steady increase in the circumference of the base of the horn, and that was good. They're just getting bigger as the animals grow. But one neat thing was that if you looked at the height of the horn, or sort of the thickness at the base of the horn in a forward-backwards direction versus side to side, there's sort of a steady increase in what I was calling height, that forward-backwards dimension, whereas width was not really changing over time. So it seems that majority of the growth going on at the base of the horns in these Triceratops specimens is actually kind of occurring, like the horns are sort of oriented forward, it's sort of occurring in this sort of a way. So it may, and I think that may have to do with sort of the way the skull roof thickens and might have something to do with how the horns actually end up orienting forward. The other thing I noticed was this very prominent difference between Triceratops horridus and Triceratops prorsus specimens. And ventral length or horn length was much greater in Triceratops horridus, or was reaching much greater lengths than Triceratops prorsus across the board. And the basal dimensions of Triceratops prorsus, though, were actually still larger. So they were growing stouter horns across the board, hands down. Their horns were shorter, but also much larger in the base. And this was interesting because people have spoken before, or had mentioned before, about a difference in length being in some of the diagnosis between these species, but I hadn't heard anything mentioned about stoutness. So I was pleasantly surprised to find these differences, and be neat to look a little more into it. The other thing, and it may seem really obvious here, was that I noticed a little bit of what seemed to be sort of a tantalizing, like hinted a growth pattern in these related to horns. And as I mentioned, I included subadult and adult Triceratops horridus specimens. And you can see that as ventral length increases, the basal circumference, and ventral length. And as this increases, we see a change sort of from an area dominated by subadult specimens into one that's dominated by adult specimens. And that happens on both of these plots. And although that makes perfect sense, it's a little bit, or it's sort of relevant because there's, like I said, there's sort of debate about growth in ontogeny Triceratops at this point. And there's a few sort of growth series that have been published, and one was put out this year, or just past year, by Nick Longrich and Dan Field from Yale. And they were suggesting that most of the horn growth, or almost all of the horn growth, occurred in the subadult stage. And so what they were considering to be adult by that point, all horn growth had taken place, and the only differences now was sort of fusion of bones in the skull. And this suggests, though, that some of that growth is still going on into the adult stage. Although we'll need to sort of refine this exactly. I thought maybe it sort of spoke against what they were suggesting for growth. Again, I ran a couple of PCAs for Triceratops. And when I used only the skulls, again, there's sort of a general separation of Triceratops horridus and prorsus here. And then in this case, I used only the dimensions of the horn core. And again, there's sort of a top-down separation between prorsus and horridus. And again, this was largely related to difference in stoutness, and that as horn length was decreasing, we saw an enlargement of basal dimensions, or as basal dimensions were getting smaller, horn length was increasing as you move up and down these axes. And that just sort of corroborated what was coming out of those bivariate plots. The other aspect that I looked at in Triceratops was shape, and I was looking at the ventral curvature of the horn. So what I did was took as many specimens as I could that had fully intact horns and in good shape, and used a program where I could sort of trace the curvature along the ventral surface from the contact with the orbital rim up to the distal point. And you sort of do this on multiple specimens, the same thing I did in mountain goats. and you end up with a equal distribution of points and use the same number of points on each specimen. And then the program will sort of look at the difference in the curvature over time. And the reason I wanted to look at that was, if you remember back when I showed you that growth series, there's sort of a steady deflection of the horns from a very upright point or upright form to sort of an anteriorly directed procurved form. And this has been suggested as an ontogenetic indicator, and I wanted to see what sort of variation was capturing, or I could capture related to that. So this is the principal component scatter it put out, and these are my sort of cut and paste renditions of the shape. And so if what you're looking at here, the orbit would have been down here, and this is the ventral surface of the horn as it curves away towards the tip. And that would have been the tip there, and then the dorsal side would have gone back this way. So the major axis here, the first axis, captures the majority of the variation. And what you see moving from left to right or from right to left is sort of a change from this sort of straight with a recurved tip to a steadily curving horn curvature. And that's basically a really good thing. It's what I wanted to try and capture. This is the variation that's thought to occur as they grow. The only unfortunate thing is that I couldn't make a comment whether or not there was any sort of ontogenetic scatter along here. And that's because I included several of the sort of isolated postorbital horns, and the only way I could actually even assign ontogeny to those was based on curvature. So to use this then to make a statement about curvature was circular. But it does suggest that that variation, or that is the largest source of sort of shape variation in the ventral curvature. And now if someone was able to apply these methods to a sample where they had a second method of aging those horns, like if you're using a large sample of fully formed skulls and could age them based on fusion patterns, then that would actually tell you whether or not there's an ontogenetic signal across here. And that would sort of corroborate that as a indicator of growth. So I was quite happy to find that as well. So based on the Triceratops work I did, it turned out that there was a lot of confounding variation, and that came from including both species and also by including subadult specimens. And unfortunately, it was sort of my hands were tied a little bit based on sort of the limited sample I had for Triceratops. And I was hoping that including those would be informative, and it was, but just not related to dimorphic patterns. Basically, the variation I did find supported the distinction between Triceratops horridus and prorsus, so that was good, I guess. And it was surprised to find that prorsus seemed to be consistently smaller and have stouter horns, although I'd like to look at a sort of a larger sample to really support this. It was kind of neat to find. As well, it seemed that, or there was a little bit of evidence that suggested some of the ontogenetic trends suggested in Longerton Field last year, maybe that some of the evidence may speak against that. So I'll need to look a little bit further and give Dan a call. Finally, the eigenshape analysis recovered the variation I was hoping to find in curvature, and this could be ontogenetic. And although I can't say one way or the other, it suggests a method that someone can use to test this as an actual growth process. So overall, I didn't find enough evidence to sort of reject monomorphism in either of these species. In both cases, I think additional ontogenetic control is needed. We're getting fairly good resolution now for stratigraphy and for taxonomy. That's starting to be nailed down quite well. So the remaining thing is to make sure that we're actually comparing adults to adults or mature specimens to mature specimens in both of these species. And that's under a lot of scrutiny in Triceratops right now, so that's yielding lots of good progress. Although I would certainly argue that it's at least as confusing, if not more, in Centrosaurus, and that that's something that could use a lot of study, I think, and could be some really interesting things going on there. But overall, then, it sort of raised the question as to whether or not it's time to start looking at monomorphism in these dinosaurs. And as I mentioned, there's sort of this expectation that sexual dimorphism is present, and we just haven't really found it yet, and that they're doing all these fun things like fighting each other and picking who's got the bigger, sexier horns or not, and a lot of sort of projection of evolutionary theory sort of onto these animals. But it may be time to start looking at what would be happening if they are indeed monomorphic. And I can't remember. Yeah, and I think the real one is Triceratops, and perhaps if indeed there is no sexual differences in these species, then that suggests that perhaps there was selection pressure that was maintaining a consistent horn form, and one of the more obvious ones would be predator defense. If males had large horns and used them to fight off T-Rex, that's great. But if you're female and don't have horns or had smaller horns, like, well, you might not be happy about that when 6-ton beast is stomping around after you. So perhaps this is a product of predator pressure or a selection pressure sort of for predator defense. There's lots to explore, but I think that it's worth sort of considering what monomorphism may mean to how these animals evolved. And then in Centrosaurus, it seems like they're sort of doing something entirely different, and the variation we see in their horns is very considerable. And I think it's rather unlike anything that I've sort of looked at when people have studied horn variation. And once we can sort of start to nail down a little bit more firmly what's going on in terms of growth and maturity, we could revisit this. But until then, it's a fairly open question, I think. So that's the work I did and my findings. Thank you very much. And there's a lot of people I needed to thank, including the staff and collections managers here who've been really helpful over the last 3 years. And to everyone for coming. Thank you.